Yeah, I know. Your nightmare just got worse. So, if a person wanted to ask, hey, how could I make these videos even worse for students to process through? Hey, put my smiling face in there. That's going to make it worse. Hello, A&P students. How are you doing? Man, it's good to connect with you this way. Yes, somebody got a new toy. It took a while, saved my lunch money up, saved my allowance up. Ain't been able to buy any Pokemon cards in about 3 weeks, but we're here, we got it, and so now I can start to get into those cards again. Hey, how you doing? Hope it's going well for you. For me, as you can see on here, it's Labor Day weekend. This is Chapter 4 stuff, so we don't have a class on that Monday, and then on that Wednesday we would have done or worked on Chapter 3. We did those. Oh, that's right, we did those questions. I'm sure that was a great time. Of course, I'm talking about it beforehand, and at that point you already experienced. But you know, I'm sure it was a great time, and whatever happened, we made it work to the best of our abilities. So, anyway, welcome. Glad to talk with you, A&P students. Glad to see you. See you, air quotes. Hey, you get to saw the air quotes that time. This is what's going to be the best part about it. I've already made one of these for the bio side, the dual credit at Nevada High School, the Crowder bio students. So although you will see yours, let me think. That was Chapter 5. Yeah, you should see this one before they see theirs. So you've got that bragging rights right there. But hey, hope it's going well for you. So I think, if I remember right, we're going to see this. You'll see this. Let's see, we did 2 days of Chapter 3. And then 2 days of Chapter 4. So yeah, it probably next week sometime. Yeah, next week sometime. But anyway, hey, glad to connect with you this way. I hope you enjoyed this. This is going to make it more enjoyable because now you get to see me do all my crazy stuff. And after doing the bio one, man, one thing I got to watch for is just, when you don't have a camera on, if your arm itches, you just give it a little scratch. If you need a drink of water, you just take a drink of water and so on and so forth. But got to watch that kind of stuff. Anyway, but with that, hey, let's get started. What we're going to be talking about today, yeah, I know the forehead's pretty bright. I may have to raise that lamp a little bit to help with that, but nothing else. Maybe it makes it even easier to watch, deal with. I think what some of the bio students did, and maybe you'll do it as well. Let me get a little pen here. I use blue for them, so I'm sure that's what's up. So let's get a different one. I'm sure the bio students did this, and so maybe it'll help you too. But get you a little 3x5 post-it note, and then just put it right over that section right there, and you won't have to deal with looking at me any longer. But hey, let's get started. Let me get that red square off of there, so it's not in the way of anything. There we go. Oh, got one more little spot there. All right, let's get started. Hey, let's pick out a good color here for us. What am I feeling? This is DNA. Does the DNA color jump out at me? You know what? How about some gold? Since this is such an important topic. Get that fine point right there. Close that out. All right. Hey, let's get started. Oh, there was my first scratch right there. I got to watch doing that. Anyway, got me a glass of water ready to go. And also I got my Chiefs coaster as well. So I am ready to go. So let's get this DNA party started. Hey, here we go, DNA. Now we did a little of this, and let's think, for you guys, did we? Oh, nucleic acids. When we talked about macromolecules. Yeah, we've done a little bit of this. So this first part will be somewhat of a review, but then what we're gonna focus on is the fine details, especially the processes, replication, transcription, translation, Oh, that reminds me. This is going to be a two-parter, as all of them seem to be that way. They're not just gargantuan in size. And in this first part, we're going to do structure, and then we're going to do transcription and translation. And based on the PowerPoint, based on the textbook, replication is done at the end, which is crazy because it has to be done first in order for the rest of these to happen. So why you talk about it third, I don't know. It is what it is, so we will follow that format. So for this first part, it will be structure, as you can see right there, the old word structure, and then transcription, translation. And that's the cool part, because that's converting it to the transcription, converting it to RNA, and then the translation is converting that RNA into a protein. And there's my first hand gesture there. Little air quotes there for you again, just to get you fired up about the rest of this lecture. So, hey, let's get started. Let's get this done. So I'm going to close this out because I want to, and actually I probably didn't need to do that, but I am going to have to move down, make this stuff a little bit bigger, and get it out from behind me, and get my pen back up and going here. And so here we go. So we're going to start off right over here, and this is very similar to what we saw in macromolecules. Give me a quick drink there. And so what we saw in macromolecules, it's the structure of DNA, and of course RNA would be the same, just a little bit different than this sugar down here, but we will see that. But the basic structure, we got our phosphate group, okay, and that is the same phosphate group that a phospholipid has. And that phosphate head that was attached to the glycerol, that was attached to the fatty acid tails, that's the same phosphate group. Phosphorus surrounded by a lot of oxygen. Okay, the second thing, and we're right here, okay, the sugar. Okay, we got a sugar right there. And of course that sugar is what determines if this is RNA or DNA. If this is deoxyribose, then it's going to be DNA, which in this one is. But if it was ribose, okay, it would be RNA. And those two are super important, and we're going to see that when we talk about this one right over here, because of what their structure is and what their part is in the whole structure of DNA. And of course, last but not least, we got our nitrogenous base up there, and this particular one happens to be adenine, which you can see here is a purine. The purines have two sections, and the pyrimidines just have one section. So you've got your A and G, you got your C and T in those. Oh, excuse me. And hey, politely ducked out of the camera way there, so you need to see what just happened there. I learned that in broadcasting school when I was doing all that stuff. So to get off camera whenever you got to do something that don't need to be seen. All right, so that is the basic structure of DNA. We got a phosphate group, we got a sugar, we got a nitrogenous base, and it can either be A, which is shown here, or it can be T, thymine. It can be C, and I drew a G, so it can be G, guanine, or C, cytosine. Any one of those three could be right there. Let me close this out and move this a little more central here. So now we're going to talk about the structure of DNA and the coolness of it. We know that it's a double helix. If you watch The Big Bang Theory, you see that model over in the back. Typically, if they're on the couch, it's in that upper right-hand corner of your screen. If they're in the kitchen and looking back into, like somebody is at the counter and they're looking back, again, it's going to be kind of in that middle back there. But there was a famous scene where it actually fell apart and they had to put it back together with glue and stuff. So, but that's the DNA structure. But we know it's a double helix. But if you could somehow untwist it and lay it flat, okay, you could metaphorically compare it to a ladder. You know, you got your side rails, and then that thing you're going across where you put your feet, the rung of the ladder. When we laid out flat, kind of like what we got here. Oh, I can get my pen up here. What we have here, it would metaphorically resemble a ladder. With this would be the side rails, and then this would be the rung in between where you put your foot as you're going up to the ladder, maybe to clean out the gutters, or you're painting the wall or something, or anything, you know, getting up there on the edge of a tree to cut branches down, whatever. It can take that shape. So the structure, we got our side rails, okay, and they are made of these sugar phosphate, alternating sugars and phosphates. And you can see that right here. And let's just go ahead and circle the whole thing. We got sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And that would be the side rails. And the same thing up here. Sugar, phosphate, sugar, phosphate, sugar, phosphate. And the stuff going across here, that's the nucleotides. That's that's the nitrogenous base. That's these guys right here. And you can see that based on the pairs, the complementary base pairs, we know that T and A match up. We know that C and G match up. And so, you know, we've got the purines. We got the 2 here, and the pyrimidines, purine, pyrimidine on that. But another cool factor is these bonds in between. Now, if it's A and T, they get 2. Okay, they get 2 bonds, and if it's C and G, cytosine, guanine, they get 3 bonds. And hopefully, when I mention these bonds in here, and the fact it's DNA being held together, that hands all over the room, all over the place, everybody's screaming at their screen, what kind of bonds those are. Okay, and if you're not remembering, let's see if we can't give some subtle clues to see if you can bring it to mind. First of all, they're weak bonds, and that is very helpful and proficient, which I've mentioned many times. Hopefully that gave you your clue. If you still don't have the answer, then let's go with it's the same bonds that hold water molecules together. Now, hopefully all of us now have it. And of course, these are hydrogen bonds. Hydrogen bonds, let me put the word hydrogen over here. Hydrogen bond. And yeah, I got this new toy. Actually, I got several new toys, but you didn't know this and you can't see it because I can't hold it up. But not only is this lamp new, the desk that I'm doing this on is new. I just needed more space. And so I told my wife, I said, hey, I just need a new desk. And she said, you don't need a new desk. I said, I need a new desk. She said, you don't need a new desk. So I didn't get a new desk. But then I told her how, but she didn't like the desk that I had. Thought it was ugly. But I said, hey, if we get a new desk, then you get rid of the ugly desk. And plus, we can sell the desk that we're getting rid of. And whatever money we use, you can use to make you a new garden. Okay, and so that sold the deal right there. And when she does make this new garden, it'll be garden 94, I believe, around our house somewhere. So, yeah, she loves her gardens and doesn't worry about how it affects the person who does the mowing. The person who does the mowing is right there. She's not worried about that. She's worried about getting the garden in. So, whether it's flower gardens, vegetable gardens, whatever kind of garden, we even got a rock garden. The rocks haven't grown very well, but we got a garden for them nonetheless. So yeah, she loves her gardens, likes the pretty vibe, everything, but again, not worried about the mowing. But anyway, so I got me a new desk, got me a new webcam, and so the next thing I got to work on is some better way of taking care of this, of writing on the screen without using the mouse. So that's my next project. So if you got an idea, let me know. That's my next project with these videos. So back to our Yeah, hydrogen bonds. These are hydrogen bonds in here. Weak bonds, but super efficient, super helpful because they allow the molecule to be taken apart, in this case DNA, taken apart, put back together very easily, very efficient, doesn't require a lot of energy, and that way the DNA structure can be copied and put back together in a super, super, super meaningful way. All right, so let me close this out. Let me go up here just a little bit and bring up these three. This one, get my pen here. This one we've pretty well already handled, but just the idea of pyrimidines are single ringed nucleotides or nitrogen bases, and the purines are double. Now, what would make it super helpful if the complementary base pairs matched up with the purines and the pyrimidines, but they don't. It should be like purines is A and T, or purines are C and G, and pyrimidines are the other set. But they don't make it that way. So what you can remember is that in every connection, and you can see it right up here, in every connection there is a two-ring structure connected to a single ring structure. But it's also best just to remember that adenine and guanine, AG, assemblies of God, if that helps you. The assemblies of God's group are the purines, and the pyrimidines, CT scan, are the pyrimidines. And hopefully the Assemblies of God people don't get upset that I use them as a purine example or a way to remember it. And if that's your religion, I do apologize if that was not considered. All right, so now we will tackle this. And again, this is going to be mostly just a review. But again, we've got the nitrogen base, the rung of the ladder, double bonds here, double hydrogen bonds, our sugar phosphates. Remember, the sugars are attached to the nitrogen base. So anytime you see a nitrogen base connected to the side rails, that's a sugar. And the phosphate groups are these little bitty yellow circles, much like up here they were a little bitty green. But you can see the sugars, okay, because they're carbohydrates, they're going to have that 5 or 6 ring structure that we saw and talked about way back in chapter, it's macromolecules. So your chapter 2, bio, is chapter 3. And of course the triple hydrogen bonds in between the G's and C's, but again a purine attached to a pyrimidine. In all scenarios, just the letters switch up. Now we're going to get into this next topic later. Little glass of water there. We're going to get into this next topic a little later when we're dealing with proteins, because as we showcase a protein being made, we're also going to talk about that final product of when proteins are folded up in such a way in the structures. And what's happening here in this slide is the same kind of idea. The DNA, we're going to walk it through. This is going to showcase the DNA being packaged in a way that takes it from being this open and this big, this section here, all the way down to a tight chromosome or sister chromatids, however you want to say it. And it's all about winding around in this manner here. And so we just wind it, and the more we wind, the tighter it gets, and the tighter it gets. But it's just the idea of folding it in such a way to where it's compact. So we got our double helix, and then we go around these core particles to shrink it down, the diameter down. We again fold it some more, the nucleosomes, and we're going to showcase that here in just a little bit. But that's this little section If I can't quite see it, but I'm going to put an arrow right there. That's this middle section right here. Again, just folding, folding, folding. And matter of fact, let's just bring this up because this is kind of the final product. This. Oh, I didn't mean to do that. Pen, please. Okay, this is the same as this, obviously. And we talk about this and we'll identify some parts, but you can see, let's go a little bit bigger here. You can see all this waviness right here. That is no, I didn't do that right. Yeah, that's what I should have done. Yeah, basically where you put the mouse, and then when you bring it, make it bigger or smaller, kind of moves it in a particular way, and I forget that sometimes. So all of this, structure, winding it up, coiling it around, condensing it, folding it in such a way it creates these wavy patterns, as you can see here, into a chromosome. So similar to how the proteins were, my eye is itching, similar to how the proteins were folded, the DNA gets kind of the same treatment there. Okay, so let's bring this up, and just to kind of show you where we are, we got these two pictures, we got these two pictures, and we got, well, actually, I'll take that back. I thought we was maybe getting close, but no, we may be halfway at best. All right, so actually let's bring this one all the way over, and let's kind of blow this one up. Get my pen back here because there's three things that we want to identify, and let's take this eraser and get rid of that one. That's supposed to be a T. All right, this one, it's got a couple of names, typical science stuff. I don't know why we got several, anyway, I don't know why I paused there. But what's going on here, this is pretty cool. What goes on here, this is the connection point to where the microfilaments connect to the chromosomes. They kind of like anchor on here, and that's how they pull it around. So if you can imagine, I got a little truck here. I don't know why I got such a big front end, but little wires here. So, and you can imagine now if I pull this forward, I can move this chromosome. And if I put one here on the back side, pulling this way, that's how we're actually going to separate it during anaphase. So they connect on there, and then when it comes time to, they either move the chromosome, because they move it to the middle of metaphase when they're all lined up. I'm sure you've seen, remember that line, where they're all lined up in the middle, and then the attachments on both sides, and they big quick torque movement, and they separate them out to each of the ends of the cells, and the cell divides, and in each set, each cell gets a set of chromosomes, or has a full set. It's duplicated, and then you got twice as many, and then when you separate them, you've got the full set there. So that is that part. Oh, the other part is just this is this whole thing. If I could say this whole thing, that's a chromosome. That's a chromosome, but individually, that's a chromatid. That's a chromatid right there. And of course, when they're together, sister chromatids because of their connection to each other. Okay, so let's bring this over. Okay, now we're going to begin the transition to talking about RNA. And so I brought this up. Once you notice, you can see a picture of RNA, which as you can see is right there, but also in a comparison to DNA. So right away, the two obvious differences is one, DNA has two parts, two strands. RNA has one strand. And the reason why that is because if I have, say this is a DNA strand, okay, and it's unwound and then they're separated. And so then they're red, each of them are red. Let's get a different color here. Use red since I just said the word red. As the polymerases read the DNA strand, it makes a copy of the nucleotide. I don't know why I'm doing that so much. And the same thing over here. And so it converts it from DNA to RNA. Okay, and then these RNAs, they leave. They go out into the cytoplasm. And then the DNA strand is put back together. So this is RNA. And they go out to the cytoplasm and as individual strands. They're not put together. No, they go their own separate ways. The other big difference is right here, uracil. Okay, on RNA, the base pairs are A and U, and then C and G. So no more T's. So when we're reading the DNA strand, if a T is red, then they put an A opposite of it. But if an A is red, then they put a U opposite of it and keep that format like that. All right, so that is the comparative structures of DNA, RNA. Same sugar phosphates, okay, same rungs across or rungs out there. It's just they're not connected to their side, but it is super important because eventually they're going to get read by some tRNA, and then a pretty cool thing begins to happen. So let's close that. Let me back up and kind of see where I'm at. So yeah, we're going right over here. All right, let me make this bigger again. Now we're going to talk about these three things, and we're just going to put stuff underneath there. So let me draw myself some lines here. Let's go back to the gold to make a little separation here. All right, now replication, that's in video 2. We're going to do that one later. But we're talking cell division. DNA gets read, cell divides when it's time. But transcription is DNA to RNA. Transcribing it, okay, converting it is probably a better word, from DNA to RNA. And then translation is RNA is translated into a protein. E-I-N. Converted into a protein. Now, the way that happens, and let me take a look at this picture down here and see if this is where we're looking at that. Now, let's talk about it first. Wait, didn't I have a... Oh no, here, there it is right there. There's what I want right there. And the way this happens is this is an RNA strand. You can see mRNA. Oh, I don't need to do that. Okay, so RNA strand. All right, that's an RNA strand. And you can see all these, you can see all of these nucleotides, nitrogen bases. Now the difference is, excuse me there. The difference is that, of course, it's an RNA strand, but now they've begun to being processed in these things called codons. Okay, a codon. Now let's type this one. Let's type this so it's really legible. Okay, codon is a set of 3 nucleotides, and you can see that codon 1, there's the 3, codon 2, there's the 3, codon 3, there's the 3. And what those do, based on what combination it is, determines what amino acid. So if we take that AUG, and hopefully you've seen a chart like this, but I'm going to walk you through how to read this, and then we'll do this in class as well, just to make sure that you are golden on it. And so it already shows us that we're going to make the met amino acid, and that is, where are you at? Oh, right there. Okay, so the way you read this is I got AUG. So I start on the left-hand side, okay, and I go down to my first letter, which is A. So I'm here. So my amino acid is going to be somewhere in this row right here. But using these 3 letters is a way to cross-reference and categorize them. So now I go to the top. So I read the first. Now I go to the top, so I know I'm in this row. And my second nucleotide is U. So now I'm in this row, and so now I go to the U column, and now I know I'm going to be somewhere in this box. Okay, and then it reads. And this one doesn't. Oh no, here it is over here. UCAG. So UCAG. So the boxes going down are. If that letter would have been a U, then I would use this first one. If it had been a C, I would use this one. But it's a G. Excuse me. So I go down to the 4th box, and that's where I pick up my amino acid. And of course, the tRNA would then go out and get that amino acid and connect it to that spot. And as we go through here, each of these codons, then we go to the second one, ACG. So we go A, and then we go over to C, so we know we're going to be in this box. And then we go down to the last one, which is G. Okay, and now we've got, this is our amino acid. And it keeps doing that until it gets all the amino acids collected. And then, as it puts a new one on there, polypeptide or peptide bond that connects 2 amino acids together to form the protein. We talked about that. We talked about that just last week on Wednesday as we were going through proteins. So polypeptide bond peptide bonds in between each amino acid, polypeptide because there's a bunch of them in a long one. But the tRNA polymerase just keeps reading along here, and they keep attaching amino acids to properly, and they just keep going until they get to this bad boy right here, the UAA, which means to stop. Okay, so once the polymerase gets to that, it says, hey, we're done here. It falls away, and the RNA strand then, the protein, the conversion to protein, just goes wherever it needs to go to, whether it's inside the cell, outside the cell, wherever it takes off, and does all of that. So that is I meant to do this. So let's get back. Let me do that. So that's transcription, translation, the idea of converting DNA to RNA, that's transcription, RNA to a protein, that is translation. So that's the idea there. And now let's look at some pictures. Now let's see. Now let's take that. We've done a few more things. Let's see what we got left. Hey, now we just got the 3 pictures left. Yeah, we're in pretty good shape. All right, let me come up here. This is a image for the reading of the RNA strand and making the protein. Now what we've got here is these are our proteins. We got one right there. We got one. Right there. And so the structure of the folding, remember we talked about that, how they're folded with primary and secondary and tertiary and quaternary. They're folded, but it's the idea of the codons come through, okay, the tRNA reads what it is and then connects the proper base pair and the proper amino acid codon there, and of course we get this forming peptide bond, and of course all the folding there and the different structures. All right, so closing out my ink tray here. And so now we've got 2 pictures. One of them is they're both actually reviewed in Nature, and then we'll be done. And pretty excited about being done. Not that I want to leave you guys, because I love doing this. I love talking about this stuff. But we got a nice juicy ham in the smoker, and it'll be within 30, 45, maybe an hour's worth of being done. Here it is, 3 o'clock on a Sunday afternoon. It's hard to imagine this is Sunday because tomorrow is Monday, and typically you have a school day, but it's really a fake Saturday, so it's to throw my whole deal off. But anyway, it is Sunday. Now let me get a little bit bigger here so we can see all of this, and get me a pen. And the words are already here, so we're just kind of going to go over this. But this is a review from Organelles. So your chapter 4. No, I just made that one this morning. No. You watched the bio. So chapter 4. No, your chapter 3. Your chapter 3. Too many chapters to keep track of. And luckily the information is easy. It's the chapters that are in. That's the struggle, which is real. Uh-oh, I'm getting low in the water. Better hurry up. Alright, so if you'll remember back and when we talked about how proteins are made, this is the process. all the way from being formed all the way to getting them out of the cell. Okay, but it all starts in the nucleus where the DNA is copied, but once that protein is made, the ribosomes, reading the RNA strands and forming the proteins, we're going to call that one right here. And then they make their way through the ER, and they get in these little vesicles. Okay, and they kind of bud off there, and and then they just travel. You can see one here, and you can see one here, and here's a big one here. They're making their way to the endoplasmic, not the endoplasmic tree, that's where they are. Making their way to the Golgi complex, Golgi body, Golgi apparatus. Oh, and here's a great example of science. We'd already encountered Golgi apparatus, which is what it should be called. But you'll see Golgi body, and now here we got a third reference to it. Come on, science people, let's get this organized. So it's in these transport vesicles, and it attaches and just basically is thrown in there. And these are all individual little proteins here, and they're just wandering through these hallways, okay, being prepared for transport. They're being packaged. Remember, the Golgi apparatus is kind of like the Postman or the FedEx or the UPS. You can take your package stuff there. They got boxes, get it all ready to go, and then ship it out. That's the Golgi complex. Okay, once it passes through all this, now it's really ready for transport. Gets in these little vesicles again, and in most cases, makes its way to the edge of the cell and exit through the plasma membrane, the membrane, after they've been checked out, regulated, and are allowed to move on. So that's the process from ribosomes all the way through exocytosis, exiting out of the cell. Okay, now for our second review, we're going to go over here. Okay, and this is more of a review of everything that we did today. And my nose itched again. But again, you can see that it's all written here for you already. So we're just going to talk about each of these. But this is start to finish. Okay, we've got a DNA double helix molecule. It gets opened up and it gets copied and read, and that takes us to this. Okay, so that's a DNA strand, and it gets copied and read, and now we're going to convert it. We convert it to an RNA strand. So that's what we got there, and then the RNA strand attaches to a ribosome. Remember those two subunits? We got the great big upper half, and little bitty second half or B subunits, and the RNA strand just goes right in here and gets read. Okay, and that's the process we just talked about. It gets read in those codons. You look at the amino acids and you get the right one, and then you build. And of course, this is a protein that is building, and it's a protein with the idea of, you can see the folding process beginning. The nose won't stop itching. See the folding process, and it forms these. You got these amino acids, and then we form through peptide bonds. We bond the amino acids together, and now we got us a protein and all that other stuff. And I can see the top of it, and I'd forgotten about this. We were going to review this. So this is a review over the proteins. So let's do this. And let's do this in a quiz review style. So I will put the word up here, and then I'll pause and give you a chance to. This nose is driving me crazy. It's just like, ugh. So then I'll give you a chance to tell what it does, and it'll give you a way of reviewing this process. So the first thing, obviously, was primary. And I mean, I will tell you, but I won't write it because it can be lengthy. But I will tell you but you have already written these down. So primary, what was the primary part of the protein structure and the folding process? So I will pause here dramatically. I lean back in my chair where you think about it. Give it a little stretch there. So hopefully you were able to recall that primary is the making of the protein strand, the polypeptide, the peptide bonds and everything. It was just the making of the strand. Okay. Secondary of all the processes. Secondary might have been the coolest. It's the one I think is it's got the most cool factor because of what it does. But secondary, what do you got for secondary? So I'm going to pause here. I'm going to take my coasters. I'm going to move them to the other side of the desk just to stall here a little bit. Now they're over there. Secondary was the idea of the pleated sheets and the folding and the twisting. You fold it up like a little accordion. You take the paper strip and you fold it once and you fold it underneath and then back over and underneath again and back over till you can take this long strip, and you can't see my full arms here, so I'll go take this long strip and you can compress it down because it folded into a very thin section. Take something very long and compress it down, but then you can also twist it as well to make it more not only concise this way but inwardly. So if I leave it flat, the diameter may be an inch, but if I twist it, now I can get the diameter down to 1/4 of an inch or something like that. So that's secondary. The third one, tertiary. And hopefully now either you have gotten the first two out of the way, and so now your choices are just two and you're pretty good on what's left, or you did the real cool thing and just flipped your notebook over to back to those pages and took a look. Tertiary. So hopefully that drink gave you enough chance I should say, to come up with tertiary. Tertiary was the idea of, and I just went back. Oh, yeah, just like what we did with the proteins. You're starting to wrap, you coil it around itself, just like you would a garden hose, putting the garden hose away, or an extension cord. And, you do the old wrap around the arm thing, and then once you get that circle, then you can connect the middle, crimp the middle down, and connect that to make it thinner. Then if you do the middle, then you can just flip it over on itself again and connect the ends. And now you've taken this big loop and you fold it in half, and then you fold it in half again to make it smaller. That's tertiary. And it's got those bond links that we talked about, the sulfide bonds and things like that. And saving the best for last. Now quaternary, Q. Quaternary, that's number 4 on the list, and that was simply taking multiple proteins that have been folded individually and putting them together to make a big collective unit, all with the purpose and design to just make them concise as possible. You're taking a week's vacation and you got 3 bags, but you can only have 1 on the plane, so you got to take all that stuff. You don't give anything out. You got to take all that stuff and condense it down to 1 suitcase. So you really got to fold and condense and pack tight and push and shove and get everything in there. Have the little lady sit on the suitcase so you can zip it up. All that good stuff. That's the purpose and processes behind the 4 levels of protein structure. Hey, there we go. We got all the way through. Let me zoom out here just to make sure I'm not forgetting anything. We're going all the way out. Did we do everything? Heck yeah, we did. Close this out. And yeah, we did everything. Good job by us. Good job by you. Good job by me. We all did a good job. Smiley faces. Where's my pen at? Oh, I don't got my pen. Smiley faces for everyone. And smiles there if you need those. maybe a little nose too. Smiley faces for you, smiley faces for me. Hey, good job. Thank you. Hope you took good care of the notes there. Lots to write down, and this PowerPoint can be and will be provided, shared with you. I'll put it on the Blackboard site. If I don't, somebody remind me, and I will get that up there. But it's got these pictures for the most part. Sometimes they won't copy over or paste over, and so I got to use different ones, but I try to make them at least as similar as possible. I think most of these came from the PowerPoint. So with that, hey, enjoy the rest of your weekend. Enjoy the rest of your week. Hope it's going well for you. Hope you're well, safe and secure, and everybody in your family is well, and that you have avoided any unfortunate situations with the virus or anything else. I mean, it's we're heading into mid-September by the time you see this, and so maybe it starts to cool off, maybe it doesn't. It is whatever it's going to do, it's going to do. But anyway, just hope you're in a good spot, you're in a good place, mentally, physically, emotionally, and you're trudging along. And if any point in time, hey, I can do something for you to help out, help you to understand something, work out something, hey, just shoot me a message. If we need to talk on the phone, whatever, we can always set that up and get that dealt with. But enjoy yourself. For me, great rest of the Labor Day weekend. Can't wait to get into that ham. It's going to be really good. All right, with that, we will say goodbye.